Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I am Dr. Muhammad Ali Hashmi from University of Education at a campus and with you regarding teaching of Introduction to Biochemistry subject. In this lecture, we're going to study about chemistry and biological importance of membrane lipids. In the previous lecture, we studied about the chemistry and importance of the storage lipids. In the current one, we'll discuss the membrane lipids. The main concepts we are going to learn in this lecture are the importance and chemistry of the following membrane lipids. These membrane lipids will include phospholipids, sphingolipids, glycolipids, sterols, and prostaglandins. We are going to discuss each one of these in detail in the coming slides. So the central architectural feature of biological membranes is a double layer of lipids, which acts as a barrier to the passage of polar molecules and ions. What does that mean, that the central architectural feature? It means the basic structural feature, mean how the biological membranes are made. That is their architecture, their structure. So they are present in the membranes. The lipids, these are present in the membrane as a double layer, which is also called as lipid bilayer, as you might have studied in the earlier classes of biology. So that double layer of lipids acts as a barrier to the passage of molecules and ions. So that double layer is there to protect the cell from everything which enters the cell. So it allows certain things to enter the cell and it stops some things which are not required or which may be dangerous to the cell. We are going to study in detail the function and structure of these lipid bilayers in the next lectures. Membrane lipids are amphipathic. That what does that mean? It means that they have both functionalities, the hydrophilic and hydrophobic functions. So their one end is hydrophobic and the other end is hydrophilic. So what does hydrophobic mean? Hydrophobic means water repelling, and hydrophilic means water attracting. So as you'll see in their structure, they have non-polar and polar parts. The non-polar parts will be hydrophobic and the polar parts will be hydrophilic. So they have a dual nature. Their hydrophobic interactions with each other and their hydrophilic interactions with water direct their packing into sheets called membrane bilayers. As we just discussed, that due to their hydrophobic interactions with each other, they form a bilayer, and that bilayer, its heads are hydrophilic, so they face the water side, and their tails are hydrophobic, so they are together, which makes a lipid bilayer in the membrane. We're going to see it in the next lectures in detail. So in this one, we are going to discuss the three general types of membrane lipids. The first one is glycerophospholipids, sphingolipids, and sterols. What are glycerophospholipids? These are the lipids in which the hydrophobic regions are composed of two fatty acids joined to glycerol. We have seen the glycerol molecule in the previous lecture. It has a 3-carbon chain with 3 hydroxyl groups. So that is the backbone of glycerophospholipids. where two fatty acids are joined to the two carbons of it. In sphingolipids, a single fatty acid is joined to a fatty amine called sphingosine. We're going to see the structure in detail in the next slides. And the sterol. What are sterols? These are compounds characterized by a rigid system of 4 fused hydrocarbon rings. A sterol is a bit more complex than the open chain lipids. So it has 4 fused hydrocarbon rings. 4 rings are fused together. They are attached together, as we shall see in the last of this lecture, where we are going to study the sterols. So the hydrophilic moieties in these amphipathic compounds may be as simple as a single hydroxyl group, like at the end of a sterol ring system, or they may be much more complex, as you'll see in these glycerophospholipids and sphingolipids. In glycerophospholipids and some sphingolipids, a polar head group is joined to the hydrophobic moiety by a phosphodiester linkage. These are the phospholipids. कुछ जो ग्लिसरोफॉस्फोलिपिड्स हैं और कुछ स्फिंगोलिपिड्स हैं, उनमें क्या होता है कि जो पोलर ग्रुप होता है, पोलर हेड ग्रुप होता है, वो हाइड्रोफोबिक मोइटी के साथ एक फॉस्फोडाइएस्टर लिंकेज के ज़रिए ज्वाइन होता है. दरमियान में एक फॉस्फेट बॉन्ड होता है, वो भी नेक्स्ट स्लाइड पे हम देखते हैं. उनको फॉस्फोलिपिड्स कहते हैं. On the other hand, some sphingolipids, they lack phosphate group, but they have a simple sugar or complex oligosaccharide at their polar ends. These are glycolipids. तो दूसरी टाइप जो है वो ग्लाइकोलिपिड्स है, जिनमें फॉस्फेट ग्रुप नहीं होता, उसकी जगह पे कोई शुगर मॉलिक्यूल होता है, उनके जो पोलर एंड होते हैं उन पे, उनको ग्लाइकोलिपिड्स कहते हैं. So here we can see that membrane lipids, if you divide them in phospholipids and glycolipids, as we just discussed in the previous slide, phospholipids may be of two types, that is glycerophospholipids and sphingolipids. So glycerophospholipids have simple glycerol backbone with 2 fatty acid chains, and then on the third carbon a phosphate group is attached, which is further attached to an alcohol. So these will be called as glycerophospholipids. The other subtype of phospholipids is sphingolipids. Sphingolipids, what is difference between sphingolipids and glycerophospholipids? In glycerophospholipids, the backbone is the glycerol molecule, while in sphingolipids, the main backbone is sphingosine. It is not glycerol. So head group is sphingosine, and then it has one fatty acid chain, and then a phosphate group, and then another moiety, which may be choline, ethanolamine, or something else. So these will be phospholipids. Then we come to glycolipids. Glycolipids, as the name implies, there should be some sugar molecule in them or a carbohydrate. So they also have two further subtypes, sphingolipids and galactolipids. Sphingolipids have a sphingosine backbone, and then one fatty acid and a mono or oligosaccharide. So what is the difference between the sphingolipids which come under phospholipid and the sphingolipid which come under glycolipid? The ones which are with phospholipid, they have a phosphate group, and the ones which are in glycolipids, they lack that phosphate group. Did you get the point? So these ones, they contain a mono or oligosaccharide. They have a sugar molecule. So that's why they are under the umbrella of glycolipids. The second one is galactolipids. Galactolipids, or they are also called sulfolipids. They have a glycerol backbone with two fatty acids similar to these glycerophospholipids. And then the third carbon has a sugar moiety mono or disaccharide with a sulfate group at the end, due to which they might be called as sulfolipids. So this one is out of scope. We are not discussing the bacterial lipids at the moment. So we first discuss about the phospholipids. Phospholipids are a class of lipids that are major component of all cell membranes. So the membrane lipids, as we started the chapter with the name membrane lipids, phospholipids, they are the major class which is present in all biological membranes or the cell membranes. The structure of phospholipid molecule generally consists of two hydrophobic fatty acid tails and a hydrophilic head consisting of a phosphate group. So, as we previously discussed, that lipids have two ends, a hydrophilic and a hydrophobic end. Phospholipids, they have a hydrophobic fatty acid tails. So you know that the fatty acids, they are hydrophobic due to those long carbon chains, and they are forming the tails of the phospholipids. And what is the head? The head group is hydrophilic because it consists of a phosphate group which is a polar group. So that is why it is hydrophilic. Phospholipids might be glycerophospholipids or sphingolipids with a phosphate group, as we just discussed here. Sphingolipids or glycerophospholipids, they are both types of the phospholipids. So now we move to the glycerophospholipids. Glycerophospholipids are basically membrane lipids in which two fatty acids are attached in ester linkage to the first and second carbons of glycerol, and a highly polar or charged group is attached through a phosphodiester linkage to the third carbon. So in glycerophospholipids, this is the molecule of glycerol. If we remove that PO3 and put a H here, that will be glycerol. So in glycerophospholipids, these first two carbon number 1 and carbon number 2, they have two fatty acids attached over here through the ester linkages by removing these these hydrogens of the hydroxyl group. And the third carbon has a highly polar group which is attached to a phosphodiester linkage. So this phosphate group is attached here, and then these oxygens are available for any other highly polar group to attach to the this phosphate group, which is directly attached to the this glycerol molecule. So glycerol is a prochiral molecule. What does that mean? That it has no chiral carbon, but just after the addition of the phosphate group at one end, it becomes chiral. So prochiral molecules are those molecules which after any change on one carbon, they become chiral. And I hope that all of you know what is a chiral carbon. A chiral carbon is the one which has 4 different groups attached to it. So if you look at this carbon number 2, it has 4 different groups. So one group is this CH2OH, second group is the hydrogen, third group is the hydroxyl, and fourth group is this whole moiety. So it has 4 different groups. That is why it will be called as a chiral carbon. So this compound can be named as L-glycerol 3-phosphate or D-glycerol 1-phosphate. If we start numbering from this carbon, it will be D-glycerol 1-phosphate. If we number it from here, it will be L-glycerol 3-phosphate. So, what is the chemistry of these glycerophospholipids? Glycerophospholipids are named as derivatives of the parent compound, phosphatidic acid. we shall see this phosphatidic acid very soon on the next slide. That is according to the polar alcohol in the head group. We'll see which alcohols are attached on the head group on the next slide. Phosphatidylcholine and phosphatidyl-ethanolamine have these are two glycerophospholipids which have choline and ethanolamine in their polar head groups, for example, and they are named according to those alcohols which are in their head group. In all these compounds, the head group is joined to glycerol through a phosphodiester bond in which the phosphate group bears a negative charge at neutral pH. We have just seen in the previous slide, look here, that the phosphate group has a negative charge. The polar alcohol may be negatively charged, neutral, or positively charged. So the alcohol which will be in the head group, that might have any charge, or it might be neutral. So here is the phosphatidic acid general structure, in which you can see that there are two fatty acid moieties on the glycerol, and then we have a phosphate group here with an X showing here. So the X might be any of these different molecules, and depending on that, its name will be here. So this table presents you different glycerophospholipids. For example, if we replace H with hydrogen, very simple, that will be phosphatidic acid. So that structure is of phosphatidic acid. And if we replace this X with the ethanolamine, as we studied on the previous slide, it will be called phosphatidyl-ethanolamine. And if there is choline moiety instead of X, so this will be called as phosphatidylcholine. Similarly, serine, glycerol, and inositol 4,5 biphosphate and phosphatidylglycerol, these can be present in place of the X, and the name of glycerophospholipid will be here. So the fatty acid and glycerophospholipids can be of any of a wide variety. So a given phospholipid, like phosphatidylcholine, may consist of a number of molecular species with its unique complement of fatty acids. So these the structure of this X will be as given in this table, but these fatty acid can vary from organism to organism and the location of these glycerophospholipids. So these two fatty acid chain, they can be different in different organisms. The distribution of molecular species is specific for different organisms, different tissues of the same organism, and different glycerophospholipids in the same cell or tissue. So that is what we just discussed, that the distribution of these molecular species that is specific for different organisms. Each organism might have its own fatty acids. Even different tissues of the same organism and different glycerophospholipids in the same cell may have different fatty acid chains. In general, glycerophospholipids contain a C16 or C18 saturated fatty acid at carbon number 1 and C18 to C20 unsaturated fatty acid at carbon number 2. That is a generality, but it's not specific. It might be different for different places where the glycerophospholipid is found. So some phospholipids have ether-linked fatty acid instead of the ester linkages. What are those? Let's have a look. Some animal tissues and some unicellular organisms are rich in ether lipids, in which one of the two acyl chains is attached to glycerol in ether rather than ester linkage. So we have seen previously, let's have a look here, that this is ester linkage. This is also an ester linkage. If one of these is replaced by an ether linkage, they will be the phospholipids having ether-linked fatty acids. So the ether-linked chain may be saturated as in the alkyl ether lipids, or that may contain a double bond between carbon number 1 and 2 as in plasmalogens. We'll see on the next slide. And where are these ether-linked lipids are found? Their vertebrate hard tissue is uniquely enriched in ether lipids. About half of the hard phospholipids are plasmalogens. So here is the structure of a plasmalogen, which has an ether linkage here and then a double bond here as well, which we discussed on the previous slide. And on the carbon number 3, it has a phosphate moiety with a choline polar group. The membranes of halophilic bacteria, ciliated protists, and certain invertebrates also contain high proportions of ether lipids. So ether lipids ये और कहाँ पाए जाते हैं? ये halophilic bacteria में पाए जाते हैं, ciliated protists में. Some protozoans which have cilia on them, they are called ciliated protists, and कुछ invertebrates में भी पाए जाते हैं ether lipids. So the next one is sphingolipids. Sphingolipids is the fourth large class of membrane lipids, and it has a polar head group and 2 non-polar tails. But unlike glycerophospholipids and galactolipids, sphingolipids contain no glycerol. So there is no glycerol present in sphingolipids. Their head structure looks quite similar to the glycerophospholipids, but they have no glycerol in them. Sphingolipids, they are composed of one molecule of the long chain amino alcohol called sphingosine. So they have the backbone of sphingosine, not glycerol, or there might be one of its derivatives. Then after sphingosine, they have one molecule of a long chain fatty acid and a polar head group. So these are 3 main components: sphingosine or its derivative, a long chain fatty acid, and a polar head group, which is joined by a glycosidic linkage in some cases and by a phosphodiester linkage in others. Let's have a look on the next slide. So, here is the sphingosine moiety colored in this magenta background, and then we have a polar head group, which is this one, the fatty acid, and an X group, which can be in a phosphodiester linkage or a glycosidic linkage. So if X is hydrogen, it will be termed as ceramide. We're going to discuss about ceramide on the next slide. So then if we have sphingomyelin, which has the same structure, and instead of X there will be phosphocholine. This whole moiety will be present here. It will be called as sphingomyelin, and that one has a phosphodiester linkage, as we studied here. And what about the glycosidic linkage? If there is glucose present here, it will be glycosidic linkage, and the structure of it will be glucosyl cerebroside. And similarly, we might have these different complex sugars at the place of X. So carbons 1, 2, and 3 of the sphingosine molecule, they look structurally analogous to the 3 carbons of glycerol in the glycerophospholipids. So if you look at these carbons, they look quite similar to the glycerophospholipids glycerol moiety. When a fatty acid is attached in amide linkage to the NH2 on C2, the resulting compound is a ceramide. So in this NH2, if one hydrogen is removed and a fatty acid is attached to this amide linkage, that compound will be called as ceramide, and it looks quite similar to the diacylglycerol. And ceramide is the structural parent of all the sphingolipids. As you might have seen on the previous slide, let's have a look again, that it is the structural parent of all the other sphingolipids. So if we put a hydrogen here, it will be ceramide. But just by replacing that hydrogen with different other groups, we can find that ceramide backbone is present there, and just by replacing a hydrogen, other sphingolipids are obtained. So that's why we call that ceramide is the structural parent of all sphingolipids. So there are three subclasses of sphingolipids which are derivatives of ceramide, but they differ in their head groups. So the first one is sphingomyelins, then neutral glycolipids, the third one is gangliocytes. So first, sphingomyelins. Sphingomyelins are classified in phospholipids because they contain phosphocholine or phosphoethanolamine as their polar head groups. So here is the phosphocholine. There might be phosphoethanolamine. So this is the phosphoethanolamine, sorry. There might be phosphocholine in place of this phosphoethanolamine, and this is in the polar head group, and this is the long tail. Sphingomyelins, they are present in the plasma membrane of animal cells and are especially prominent in myelin. What is myelin? That is a membranous sheath that surrounds and insulates the axons of some neurons. So you know that neurons are the cells which are responsible for the communication between the cells in the body, and it takes the nerve impulses from the brain to the whole body. So the sphingomyelins are present in those neurons as well. That is why their name is sphingomyelins, because they are in the myelin, which is a sheath that protects the axons of neurons. So here is the structure of sphingomyelin. If you look at its structure in detail, that's how it will look in the 3 dimensions, and it's not as simple as it looks here. Chemistry can represent chemical compounds in 2 dimensions like this, but they are actually in 3 dimensions. They don't sit like that compound on the previous slide. So these 2 fatty acid chains will be protruding out like this, and this will be the polar head group. So now you understand why we call this a polar head group and these ones as non-polar tails. You can see that these all the black color represents the carbon here. So these 2 are looking like tails, and they are hydrophobic. And this is the head, and it has polar groups present, so it will be hydrophilic. So that is the structure of a sphingomyelin. What is the biological significance of sphingomyelins? The membranous myelin sheath that surrounds and insulates many nerve cell axons, that is particularly rich in sphingomyelin, suggesting its role as an insulator of nerve fibers. So that is its first biological importance, that it works as an insulator in the nerve fibers. And it has been discovered that sphingomyelin plays a significant role in cell signaling pathways. So when cells send signals to each other, in those pathways, sphingomyelins have an important role. The second types of these lipids is glycolipids. And neutral glycolipids, they can be divided into cerebrosides, globosides. First one is cerebrosides. Cerebrosides have a single sugar linked to ceramide. We'll have a look on the next slide. That their parent structure is basically ceramide, but they have a single sugar linked to the phosphate group. And those with the galactose are characteristically found in the plasma membranes of cells in neural tissue. So if that sugar is galactose, those cerebrosides are found in the neural tissues plasma membranes, and if the sugar is glucose, and then these ones are found in the non-neural tissues. And what are globosides? They are neutral glycosphingolipids with 2 or more sugars, usually D-glucose, D-galactose, or N-acetyl D-galactosamine. So globosides have more than one sugar, and cerebrosides have just one sugar attached to them. Have a look here. This is the same table where we left these two for discussion. So these are 2 sugars. If there is only one sugar, it will be a cerebroside, and if there are 2 or more sugar, it will be if there are 2 sugars, it will be a globoside. So if it is one sugar is glucose and the other one is galactose, which are present on instead of this X, that will be lactosyl ceramide. So these are the basic structure of some cerebrosides and globosides. Next, we move on to the third type of ceramides, that is gangliosides. Gangliocytes are the most complex sphingolipids, as you shall see very soon on the next slides, that their structures are very complex. They have oligosaccharides as their polar head groups and 1 or more residues of N-acetylneuramic acid, which is represented with this symbol, and it is often simply called as sialic acid, and they are present at the termini. Let's have a look at the previous slide. So these different moieties, they are not as simple as they're looking. We'll see their structure on the next slides. They are present here in case of gangliosides. And sialic acids, that gives gangliosides the negative charge at pH 7, and which distinguishes them from globosides. So this is the structure of a sialic acid, which has a negative charge, which is present at the neutral pH, and that is why they are distinguishable from the globosides. What is a biological significance of sphingolipids overall? In humans, at least 60 different sphingolipids have been identified in cellular membranes. So you can see that how many different types of them are only present in human beings, let alone the other organisms. And many of these are especially prominent in the plasma membranes of neurons, as we just studied about sphingomyelins. Some are clearly recognition sites on the cell surface, but a specific function for only a few sphingolipids has been discovered thus far. So there are so many of them, but there is still room to study about them in detail. So we only know a few sphingolipids whose specific functions are discovered. The carbohydrate moieties of certain sphingolipids define the human blood groups and therefore determine the type of blood that individual can safely receive in blood transfusions. So the carbohydrate moieties, as we have seen on the previous slides, like glucose, galactose, and other moieties, they are actually specific which are related to the human blood groups. That is not the scope of this lecture, but just as a biological significance, you can see that human blood groups, they are identified based on these sphingolipids. So let's have a look briefly on the human blood groups, the O, A, and B. These groups are determined in part, so that is one part of the determination of the blood groups by these oligosaccharide head groups, the ones shown in the blue color in these glycosphingolipids. So if O antigen is present, the structure of these oligosaccharide is this: glucose, and then a galactose, and then an acetylneurominic acid, then a galactose and a fructose moiety. So this whole oligosaccharide is present in case of the O antigen, and in case of A antigen we have an extra an acetylneurominic acid here, and in case of B antigen the difference is this whole moiety is same, but we have galactose at the end instead of neurominic acid. So these are three different antigens which are present in case of different blood groups. So gangliocytes are concentrated in outer surface of cells where they present points of recognition for extracellular molecules or surfaces of neighboring cells. So they are present in the outer surface of the cells, and they recognize different molecules which need to enter the cell, or they even recognize the neighboring cells. The kinds and amounts of gangliocytes in the plasma membrane change dramatically during embryonic development. So when an embryo grows into an individual, slowly the amounts and different kinds of gangliocytes in the membranes of the organism, they change very dramatically and rapidly. Investigation of the biological roles of diverse gangliocytes remains fertile grounds for future research. So these biological roles are not very well understood, and they present a very fertile ground for future research. If some people are interested in going to the biochemistry, they might find this field for their future research, which has still available space to be discovered. Now we move to the degradation of phospholipids and sphingolipids. Degradation means breakdown. Most cells continually degrade and replace their membrane lipids. For each hydrolyzable bond in a glycerolphospholipid, there is a specific hydrolytic enzyme in the lysosome. So to break each hydrolyzable bond, we have a specific enzyme in the lysosomes of the cell. So lysosome is an organelle of the cell which has enzymes, hydrolytic enzymes, mean to break these hydrolyzable bonds, and each bond has its specific enzyme. Phospholipases of the A type, what are phospholipases? They are the enzymes. They remove one of the two fatty acids, producing a lysophospholipid, and then lysophospholipases are another enzymes. They remove the remaining fatty acid. Have a look here. Phospholipase A1, it will hydrolyze this bond and break down this fatty acid chain from here, and then another type of phospholipase, that is A2, that will hydrolyze this bond, and phospholipase C will hydrolyze this phosphate bond, and phospholipase D is responsible to detach this sugar moiety. So every enzyme has a specific structure and it performs its own function only. So next we move on to sterols. Sterols are structural lipids present in the membranes of most eukaryotic cells. The characteristic structure of this fifth group of membrane lipids is the steroid nucleus, consisting of 4 fused rings, 3 with 6 carbons and 1 with 5 carbons. We'll have a look at it on the next slide. The steroid nucleus is almost planar and is relatively rigid. The fused rings do not allow rotation about carbon-carbon bonds. So if you look at this structure, 3 rings are 6-membered rings, and 1 ring is the 5-membered ring. And because they are fused here, look at these are the fused walls. That fusion makes it quite rigid and does not allow rotation about these carbon-carbon bonds. So it has more or less quite planar structure. It cannot rotate and make different other shapes. And the chemical structure, the rings are labeled A through D. You can see A, B, C, D to simplify reference to the derivatives of the steroid nucleus. And the carbon atoms are numbered in blue color. So these blue color, you can see that these all carbon atoms are numbered from 1 to 27. The carbon 3 hydroxyl group is the polar head group that has a hydroxyl group here. For storage and transport of the sterol, this hydroxyl group condenses with a fatty acid to form a sterol ester. So that is the only polar part in the cholesterol. This is the cholesterol molecule, and we are studying the sterols here. Have a look at cholesterol in 3 dimensions. So that is the structure, which you can see on the right side in motion, and this is the fatty acid chain, and here is the polar moiety. So this end is polar end, and all the rest is the hydrophobic end. Cholesterol, the major sterol in the animal tissues, that is amphipathic. As we just studied, it has a polar head group, the hydroxyl group, and a non-polar body. And similar sterols are found in other eukaryotes, like stigmasterol in plants and ergosterol in fungi, for example. So in fungi and plants, cholesterol is not present, but it has its similar siblings like stigmasterol and ergosterol. Bacteria cannot synthesize sterols. However, a few bacterial species can incorporate exogenous sterols into their membranes. Like some bacteria, they can take up the exterior sterols and take up into their bodies, but they cannot synthesize it by themselves. What is the biological significance of sterols? In addition to their role as a membrane constituents, the sterols serve as precursors for a wide variety of products with specific biological activities. So they are precursors for preparation of a wide variety of products. Steroid hormones, for example, are potent biological signals that regulate the gene expression. Steroid hormones are very important in the body. Similarly, bile acids, they are polar derivatives of cholesterol that act as detergents in the intestine, emulsifying dietary fats to make them more readily accessible to digestive lipases. So bile acids are the acids which are actually derivatives of cholesterol. They emulsify the fats in the food that we eat, so that those fats are more readily accessible to the digestive enzymes, which are called lipases. Next, and the last one, is the prostaglandins. Prostaglandins, they contain 5 carbon ring originating from the chain of arachidonic acid. We'll have a look at the structure of this arachidonic acid. Their names derive from the prostate gland. Why are they called prostaglandin? Because they derive from the prostate gland, which is the tissue from which these were first isolated by Bent Simmelson and Sune Bergstrom. 2 groups of prostaglandins are originally, they were defined PGE for the ether-soluble prostaglandin, and PGF for F is for phosphate in Swedish, which are buffer soluble. Each group contains numerous subtypes named PGE1, PGE2, and so forth. So here is the structure of arachidonic acid, as we just discussed on the previous slide. That is their basic structure, and here fusion occurs at this position to make a 5-membered ring. Then prostaglandin is formed from this arachidonic acid. So I think this is pretty understandable that a fusion at this position makes a ring here. The rest of the structure is similar to the arachidonic acid. So that forms a prostaglandin. Prostaglandins act in many tissues by regulating the synthesis of the intracellular messenger 3' 5' cyclic AMP. So this is an intracellular messenger which takes messages between cells. So its synthesis is regulated by prostaglandins. And because CAMP mediates the action of diverse hormones, the prostaglandins affect a wide range of cellular and tissue functions. That is self-explanatory. Some prostaglandins stimulate contraction of the smooth muscle of the uterus during menstruation and labor. So prostaglandins have importance in the body. Some prostaglandins also affect blood flow to specific organs, the wake-sleep cycle, and the responsiveness of certain tissues to hormones such as epinephrine and glucagon. So some prostaglandins also elevate body temperature when we have fever, and they cause inflammation and pain. They are also involved in these things. Here is some interesting reading for you. I'll leave it for you to read and understand that how biochemistry acts in medicine, and what is its importance in practical life. Just have a read through it, and if you have any questions, you can post in the comment section, or we can discuss them in the meeting. And it's not a special assignment for you. Actually, you should go to this link and see different three-dimensional structures of lipids to have greater understanding of how their structures actually look like in 3 dimensions. So that was the end of the lecture, and if you have any questions related to this, you can ask in the comment section, and we can even discuss them in the Google Meet when we have the meeting time of our class. So thank you very much for your attention, and inshallah we'll meet in the next lecture. Until then, Allah Hafiz.